Okay, let us start now. Thank you so much, our beloved Dr. Manoj Maghani. You gave a very wonderful session for us. We have get a lot of information from your valuable session. Now we are going to the international session by English language. Then good morning, good afternoon, and good evening to all my beloved doctors and homeopathy lovers. Now we are here gathering together to attend our 1522nd day session conducted by International Forum for Promoting Homeopathy. This is our 1 hour session. After this session, our Malayalam webinar will be started at 9:30 to 10:30. First of all, I introduce about myself. My name is Shalu Rajan, our steering committee member. Today is our session's moderator. Okay, before this session, we want to take a 1 minute silent prayer. Please be silent, dears. Then today's our guest is our beloved Dr. PRAKASH RAO, who has retired as a research officer from CCRH by Government of India. He has 30 years experience in researching field and he has undertook the projects with UNICEF and AIDS Control Society. It's a very valuable thing that Honorable Dr. PRAKASH RAO has done the most research in our IFPH English department. He has come here as part of the 1500 celebrations of our organization and he has shared his experience as per his research perspective. Today, we are waiting for his valuable words about the topic of scope of homeopathy for the humans, animals, and plants. Now, we warmly welcome to Dr. PRAKASH RAO to this platform. Good evening, Doctor.
Our guest is about to join due to Diwali celebration. He has not joined, but he told me that he will be joining shortly. Anyway,
Okay.
It's really very nice to have you in this forum, and as well as in this forum to moderate our English sessions also. Because what was your experience when you conducted the students forum?
It's to me.
Yes, yes.
It was very nice that because I am one of the motivation speaker. Uh, Now I am researching for mind, how it's mind in our life by health or wealth or relationship. But I'm so interesting for this homeopathy and uh, previous years I was conducted our students group. A lot of informations I got from there. We will be continue this session, student session soonly. It was very nice experience. Our guest is joining now.
Our guest is joining. Shall I make you host also?
Prakash Doctor, are you
Yes,
here?
yes, yes. Let me make sir. Yes, sir has joined now. Yes, you can invite.
మాట్లాడు నేను
Sir,
Yes, yes, yes.
Yes, Doctor.
can you
Yes,
see my slides, sir?
yes, visible, visible.
Okay, sir. Yes. Time, sir, I am sharing the slides, sir.
No,
2 minutes, sir. Just sharing the slides.
Okay, you can take the time, sir. No problem.
Please, please.
Okay, okay.
ఏం కష్టపడుతున్నారు ఏం కాకుండా
ఇక్కడ ఒక్కొక్క స్లైడ్ ఇట్లా ప్రెస్ చేసి చేయి.
ఎక్కడ స్లైడ్ ప్రెస్ చేసి
ఆగు ఆగు మౌస్ ఇస్తా. ఆగు దాంతో ఏం చేయకండి.
హలో కెన్ యూ సీ మై స్లైడ్స్ సార్
No, sir. Yes, sir. Yes, sir. It's visible now.
నౌ విజిబుల్
Yeah, yeah.
ఓకే సార్ నెక్స్ట్ స్లైడ్ ఓకే
Okay, you can start, sir.
సార్
Yes, sir.
ఈజ్ ఇట్ విజిబుల్ సార్ నౌ
Yes, yes. Visible,
Good
visible.
evening to everybody. I am very sorry I am late by 8 minutes. So I made you to wait. My dear sir, today topic is scope of homeopathy for humans, animals and plants, a research study. In this research study, I want to cover all the three subjects, but was not in a position to cover because it is coming to various slides. So I made that only agro homeopathic slides, whatever the research, whatever the knowledge we gained, and whatever the research we are undertaking on mango plants. Now I am sharing one of the research study about the cotton plant and scope of the homeopathy. See, as a researcher, I want to give you the main important thing is the history of the agro homeopathy. So one of the first studies in the field dates back in 1995. Before that, in 1986 itself, see, this was internationally recognized in 1995 and carried out by a couple, Eugen and Lily Kiskul Kolisko, based on the theories of Rudolf Steiner about the body dynamic agriculture. This was internationally reputed in 1995, but in India, in 1986 itself, the studies have been started. Since then, a lot of studies have been done in Cuba, Italy, South Africa, and Brazil. The Federal University of Viscos and Minas of Brazil is pioneer in the studies. Countries like France and India. Now India is the topmost country to carry out in research in agro homeopathy. See, V.D. Kaviraj, a Dutch homeopath, he was trained in Dutch, but basically he is a Maharashtrian. He belongs to Maharashtra state and pioneered agro homeopathy. And he has written a book, Homeopathy for Farm and Garden. After his tremendous work, he has done lot of studies. He used it directly. Clinical application was done by him over the plants and over the germination. So this was his study. But in 1986, Dr. Kaviraj has used belladonna successfully to treat a disease on apple tree. He opened door for agriculture research. He preferred lower potencies because it was the starting experimentation. So he used lower potencies. but later the lower potencies have been converted into higher potencies, and the higher potencies were also found to be highly effective. In my previous presentations, I told that any medicine will work in any form of one of the potency, because it is the energy that is being carried out by the drugs, and it is not the medicine quantity in grams or milligrams that will be carried out. So the energy form of the medicine will be starting from 1x to 50 millismol potency, 0 by 1, 0 by 2, 0 by 3. So any medicine will work in any one of the form of the potencies. So sometimes some of the very good medicines, very what you call the polychrist remedies, they also work in different potencies. Because the symptoms that have been produced by the drug are in different potencies at different intervals in different human beings. So each symptom of the human being has been carried out and put into materia medica. So when we select the drug in a particular potency, that potency may work, may not work. He may need, that particular patient may need higher potency, or he may need lower potency. But usually almost all the drugs will work only on any one of the potencies, whether it is lower or higher, all are effective. The study tests present are showing that the homeopathic medicines are highly effective in every potency. So it is very difficult to assess in which potency they will be useful. So Kaviraj has used previously in lower potencies. Initially, the selection of the drugs for the plants was very difficult, because he started his research studies. He clinically applied without doing any research studies, but they were highly successful. So in those days, it was very difficult because there was no literature present in agro homeopathy. Initially, the drug selection to treat plant associated disease was based on assumptions. See, a physician used to assume something, and he used to prescribe, he used to apply the drugs in different forms, particularly in biochemics, particularly the batch for our remedies. Initially, research tried to find out symptom similarities, and plants usually don't have nervous system or arteries or blood, but they have a system of neurological communication. They communicate, they are intellectuals, and plants will have mind, but at the same time they convey their symptoms. So it is a kind of observation and a kind of pathological prescription which was found to be highly effective. Due to progression of agro-homeopathic research, now it is much easier to select a drug to treat specific plant associated with specific disorders. Drugs, what are the forms of the drugs are? Drugs must be diluted with water and mixed properly before the application. Drugs can be diluted with water at various ratios: 1 is to 1000, 1 is to 200, 1 is to 500, 1 is to 1000, and 1 is to 5000, depending upon the potency, the one drop of the medicine, and 100 ml of water, 200 ml of water, 500 ml of water, 1000 ml of water, as we like. So this is the dilution. It can be sprayed, the water can be put to roots, and so it was found to be highly effective. I will show you further research study how it has happened. So the scope of homeopathy, for it was reported that higher dilutions there is no effect of vehicle alcohol. So only the effect lead to compound drug can be studied. Sukul Rana has conducted with his team. Sukul Rana is a popular zoologist, and he is now retired. It seems his health is not good. That he is from Bihar. He used to use 1 is to 1000 ml of dilutions to antidote the effect of the alcohol eliminated, but the compound of the homeopathic drug was active. This compound of the homeopathic drug was active. Previously, in my presentations, I told the cell communication, atom communication, and protons electronic communication to each other, how the cells will communicate the disease, how the cells will communicate the therapeutic effects when they take energy, when how the mechanism of cure is being takes place. It is immunologically totally understood. Now it is highly related with the quantum physics. Quantum physics does not mean that it is a physical biological medicine. The quantum physics also takes place in biological bodies such as plants, human beings, and animals. So when the atom is charged with the energy, and that energy will be communicated to cell. This was carried out by us in Jipmer Hospital on immunological status in human beings. The same theory is applied for the plants. Here, many people have written that the plants will have energy, that will have energy biodynamics. No, it is not like that. It is purely pathological succession of the treatment. Once the cell is charged with the dynamic process of the drug, and it communicates to other cell, and that cell also will get the energy. By that energy the disease will be expelled. Whatever you think that the homeopathic aphorisms, where the higher the activity of the drug will eliminate the disease, these are the layman words or non scientific words, but there is a scientific aptitude, scientific application of immunology on a greater level, and it is very easy to understand the mechanism of action of the medicines of homeopathic system. And this will be done very easily on the plants. That was studied on human beings on immunological basis. The communication, the therapeutics, the pathways of the medicines on immunologically, what was said in the organon of medicine is true, and it is only the disease will be expelled, and the recovery process how it is taking place, that also can be studied, and that was observed. So, what was the first agro homeopathy was germination of seeds is the most important preliminary step for any crop production. The crop has to be produced means the germination. The homeopathic medicines not only affect the cure the disease, they also have enormous effect over the DNA, dinucleo DNA process, and nucleic acid. And it was also studied in vitro studies. The agro homeopathy started with observing effect of homeopathic drugs on seeds and germination of different crop species. So here we have tested, we have been tempted by the studies that have been carried out on cotton seeds. Positive effect of the drugs on plants was confirmed by various biochemical assays. See, when we have done the biochemical assays, the positive, what are the biochemical assays? The plants change in potassium, sodium, phosphorus, and other chemicals, particularly selenium, magnesium, and all the chemicals. And the disease process, when the plant has been affected with fungal infection, the fungal, the bacteria, and the virus that have been identified, and the medicine has been applied. And this also in two-way process, one sided, one is placebo compared with placebo and with the medicine. So the enormous effect you have been shown. See, the agrohomeopathy in seed germination and development. What are the process of the research? See, I am here talking about the research. See, the process of research is agrohomeopathy in seed germination and development. Agrohomeopathy in control of biotic stress. What is the biotic stress in plants is there are various causative agents for plants: diseases from fungus, diseases from bacteria, diseases from virus, and diseases from insects. These all will be known as biotic stress. And first is germination. Germination means the study of genetics, and particularly deoxyribonucleic acid, and agrohomeopathy control of biotic stress. The third content is abiotic stress. Abiotic stress means salt stress, heat stress, cold stress, drought, lack of water stress, and metal stress, that is copper, nickel, lead, cadmium, arsenic, all toxicity, sulfur, phosphorus, these all toxicity tests were carried out and studied out. And a preliminary study on effect of homeopathic drugs on plants was undertaken by Central Institute for Research on Cotton Technology and CCRH, Mumbai. So the Central for Cotton Technology, Research in Cotton Technology, is a parallel body like Central Council for Research in Homeopathy. So they want to carry out some of the studies. When they carried out simultaneously, we have taken to cope up with them the studies on mango trees and the fungal infection to mango trees and fungal infection on mango fruits. At the same time, the cotton seed germination was also studied by us for genomic study. What is the seed germination? What is the effect of the germination? So that was also the material, and a cross study was also done. It was found that the homeopathic drugs influence genetic activity and agriculture plant bodies in a big way. What happens in the DNA has been affected, whether the plant got the strength, and the plant itself never died. For example, in agro homeopathy, the control biotic stress and abiotic stress. The abiotic stress is because of the environment and because of the deficiency of the salts or the excess salts, particularly arsenic and sulfur, and particularly copper in red sand, and biotics are fungus. So the plant has withstood because there was a change in the DNA, and there was a strength to the plant. Apart from this, the medicines which will produce copper, nickel, lead, all these were used. So the plant became more powerful and vigorously has grown. I will show you later how this. So now the fundamentals you have to understand is agrohomeopathy is germination of the seeds development, a control of biotic stresses and control of abiotic stresses. So, preliminary study on effect of homeopathy was carried out by the Institute of Research on Cotton and Technology, Mumbai. So, since there is neither brain nor any, what is the theory of carrying out these studies is there is neither brain nor any central nervous system in the plants. Plants don't have brain, plants don't have central nervous system, but plants have mind. They react. See, whenever a human being passes by through them, they will touch the feet of the human being, they will have a contact with human being, they will have contact with animals, they also regurgitate, they also will express their emotions that there is some mishap or accident is going to happen, somebody, some animal is coming to eat the plants. So they communicate through their leaves and through their the kinetic energy to the other plants that some of the accidents or some of the threatening is coming from outside. So be careful and protect themselves. So these all have been studied and noticed with data. So under this background, research study on current subject is undertaken. It is found that medicated water carrying homeopathic drug and contains intramolecular electric charges, and the placebo water that was applied to the roots and at the same time is it was sprayed. So what are the changes to find out in genetics as well as the plant growth? So this phenomena is explained with the help of experiments on cotton. Many studies have been carried out. Cotton, wheat, Bengal gram and mung bean. Mung bean, the gram that is the mung. So there are theories that have been observed. One is when the drug in potentized form has been applied to the plants through water, the diluted water after giving forceful succussions. The changes that have been noticed are resonant phenomena. Previously, when I presented my lecture on quantum physics, and the cell to cell communication will takes place on 3 to 4 forms. One is resonance phenomena. That cell will, even in human beings and in plants, this whatever I am here presenting you, not only applicable to agrohomeopathy, this is also applicable to human pathology where the disease is present. There is resonance phenomena. Each object, which also include an electron, a molecule, as well as a gene, has its own natural frequency of vibrations, oscillations, called resonant frequency. This resonant frequency, when we apply homeopathic medicine on human beings or the homeopathic medicine for plants, this resonance will take place even in animals. That's why the animals veterinary, when we have done experiment to increase the milk, has found to be successful on cotton seeds and on mango fruits, and disappearance of black spots over mango fruits has shown greater effect. And in human beings also, you have seen various types of cures for various disease conditions. The only thing is the reproducibility is not possible, because number one is the proving of the medicines. We have ourselves chosen a pathway of similia similibus currenter. That means selection of the same symptoms which have to be applied to the human being, plant, or to the animal. So when we framed our own rules, the rate of curativeness will come down. Everybody says that the homeopathic medicines are lesser than p value of 0.05. It is not the lesser than p value. The p value is not applicable to homeopathic medicines, because the homeopathy is having a principle of similia similibus curanter. The symptoms have been produced in human beings in less than percentage of p value. Suppose if the 60 provers have been took medicine, and as provers, though around 5 to 8 people have produced the symptoms. Among these 5 to 8 people, one person has produced all 8 symptoms, and the other persons might have produced 2 symptoms or 1 symptom each. So when these biostatistics were taken out, the p value that is generally applied to the medical biostatistics is not applicable to homeopathic study, because it is a phenomena, it is a resonance, it is the action that takes place individually. So the medicines which have been proved individually will have reflection over the individual treatment. So when the individual treatment is taken place, it will not come under the biostatistics of p value. It is above p value, in the sense each case will be cured in each different way by each different medicines, because the selection of similar medicine is toughest task. The parameters yet to come out, we are taking out research. Now the pathological parameters in plants have been proved, and human beings also have been proved. We have studied immunologically the action of the medicine of homeopathic medicines, the pathways of homeopathic medicines, but we haven't found out the parameters are key points, or biologically, arithmetically, our algorithms are not prepared how to prescribe a homeopathic medicines on easiest way of basis. So that is always, we have to depend on our individuality, our observation capacity. Still we are highly dependent on our mental capacity. Still the algorithms have not fully developed. We developed up to certain level, we developed up to certain pathological level, but the easiest way of prescription is not possible, because the algorithms have not prepared. We have our own algorithms like repertorization. It is a mathematico mechanical process of biological science. We will take the symptoms of the biological science, we will convert them into rubrics, then we will study the repertory, then we will apply the knowledge of organon of medicine, then we will give the medicine. This is also a way of algorithms, that but it is a tough task, a hard work. So we want to make it easy. Now within 6 months to 1 year, we may also come with easiest prescription on individualized basis, because the plants, the animals which don't have expression capacity, the plants don't have brain but have mind, the animals don't have so much of capacity of mind to rethink, but at the same time they have way of their expressions. So basing on this, the pathology for plants and the pathology of animals and the pathology of human beings is one and same. The Krebs cycle is same, the physiology is same. So in that, the resonance phenomena has been noticed in the vibration and oscillation. This is known as resonance phenomena. One of the first starting phenomena of a cell or a molecule that is present with life. It is not possible without life. We cannot treat dead bodies, but at the same time, we can treat only healthy human beings and healthy plants that have disease. So this has to be taken into account. So the object that contains electronics molecules will contain the gene, and the gene that is known as frequency variations, vibrations will take place once the medicine is applied. Number two, when the bandage of an object with its surrounding is diluted and repeatedly pushes, it absorbs the energy from each successive push, that is molecular energy, which is, see, if we take up the molecular energy, the each oscillating will have the resonant frequency more and more. That is, the succession of potencies is more and more, and the potentization will increase the energy. The energy that is produced in 0 by 1 potency and 1x potency are entirely different. So the patient or the plant which needs 1x potency, 30C potency, or higher potencies is not understood by the physician, because the energy modules are entirely different in a diseased plant, in a diseased individual, and application. The energy doesn't mean that highest energy will have maximum effect. It is the only chemical that will produce, the presence of chemical that will produce the side effects. It was found that the 1x repeated doses, 3x repeated doses, 6x repeated doses of the dilution will produce the side effect in human beings, whereas in potentized medicine, if the correct medicine is applied, it only cures, and rest of all the potencies more than 12x are not having any side effects. This was proved with data. So selection of the remedy is the toughest task. Once the individual remedy has been selected basing upon pathology or individual symptom, it is going to produce the frequencies and modulation. The molecules of the drugs are subjected to successive dilutions and vigorous shaking. The bonding between the molecules themselves is within molecules, and electrons are weakened. When the electrons are not charged, the molecules are charged, the molecular energy will increase. That energy is a kinetic energy that will be communicated from one cell to other cell, which is having nucleus with life, and the cure will take place. This needs little knowledge of quantum physics, the theory of the physics, and theory of cell culture. So, without that, if a research paper has been presented in front of a normal practicing physicians, it will be little tough to understand. So, these all are research studies on account of loose bonding of vigorous shaking. Molecules, also electronic within themselves, start oscillating. Oscillating means movements. The electrons will move in a cell own individual resonant frequencies. The diseased person will have own individual frequencies when we apply when the disease is present, the disease also will produce oscillations. At the same time, when the cell has been given the support of the drug also create oscillation, and this oscillation has to dominate and give support to the human being, to the kinetic energy. So that kinetic energy, that is known as a vague word in homeopathy, which is highly scientific, which is highly respected, is the vital energy that will be stimulated and pushes the disease outside. With each push of size, swing will increase. So this is the oscillation. Then comes the modulation. This makes, when the oscillation starts, the electrons to act as a carrier. What happened? The molecules are charged. So electrons are decharged. The electrons will shake the kinetic energy into them, and they will act as carriers of oscillation of molecules and get superimposed. These all are superimposition oscillations. These are bio mechanical energetic words, so you kindly understand this. See, oscillations will takes place in molecules. Molecules are charged. These molecule will give the energy to the electrons. Electrons will become carriers, like a goods taking lorry or train, which will take the goods. This is known as regression, progression, and see application. And one by one, I will tell you. After the oscillation, the modulation will takes place. After the modulation, the transfer of energy will takes place. Weakening of the bonds between electrons. The electrons will not have any bond, that weakens, but the molecules are charged. Those molecules will superimpose the electrons, and that will be known as memory power of the electrons. They will keep the energy memory, and they will work as carriers, and they will give that memory to the other cell electrons, and those cells will carry out the medicine effect. And now the common question everybody will ask here is, beyond 12th potency, molecules of original drug disappear. So how this will be carried out? The molecules will disappear after 12th potency, but the electronics never disappear. The electrons, protons, neutrons, they are always present even in water that is charged with the energy. When the oscillation took place, the molecules beyond the 12th potency have given their energy to the electrons, and once the electron memorize the potency or the power or the energy that took place with the oscillation, that will be transferred into energy, and that communication will take place from one cell to other cell, and this communication of kinetic energy is known as the pathways of the drugs. And further dilution will increase the energy of the electrons more and more. When the molecules have disappeared after 12th potency, when the 12th oscillation has been given, when the dynamization has been carried out, the electrons are charged. The molecules will disappear. The charged electrons will communicate their kinetic energy to the electron. This is known as deployment and redeployment. In the sense, they take back, they give back, and they memorize. This is the immunology word, deployment and redeployment of the cells. So this was not understood by anybody until the quantum physics has accepted this. Where previously immunologically this redeployment and deployment, memory and rememory was studied in immunology, but that was also not considered. But when the disease has been stopped by giving vaccination, the memory that vaccine cells keeps for lifelong time, and they react with the same type of germination by virus or bacteria, then it is understood that deployment and redeployment immunologically takes place in the cells in the form of electronics and not in the form of molecules. So here again the theory of vital energy is absolutely true, but that was pathologically not studied. Now we have made enormous studies on immunology in our Jipmer hospital, and this is the outcome of pathways of activation. Then one is resonance, second is transfer of energy, third is activation. When the electron is activated, it will accelerate. See, these all are technical terms. Kindly bear with me. For accelerating rate of germination of cotton seeds, medicated water containing, see what are the medicines that have been taken up in cotton seeds germination. What are the medicines? One is abrotinum, second is beritacarb. Why beritacarb? Because stunted growth, not flowering, not blooming up, and the soil that has been without any copper or without any phosphorus. Here you have to understand the curing the disease needs medicine, but the general health comes to our diet. In the same fashion, the vitamins, the diet contains vitamins. The diet contains all the materials, all the chemicals that a human body needs. That is known as the chemicals are the proteins that we need. That is entirely different. Though the diet has been taken properly, the disease is entirely different. That will affect the pathology. The diet will have a secondary effect over the health of the human being. If, in, for example, anemia, deficiency of the vitamins, lack of strength, neurological problems, so they need diet. In the same fashion, plants also need diet from the soil. If the soil is weak, the stunted growth will take place. So apart from fulfilling, suppose if a human body don't have any deficiency disorder, if we give medicine, the improvement will be very good and improvement will be fast. But he is having anemia, deficiency of vitamins, and deficiency of the proteins. In that, he got a disease, suppose allergic dermatitis or chronic bronchitis. So, sending the disease will become a pathologically very tough, because the kinetic energy that has been produced from that human being will be very less, and that communication through electrons will be very tough. In the same fashion, in plants also, the electrons which are charged actively, when the plant pathology and the plant structure and the plant soil is very weak, that energy will be very less, and increasing the energy of electronically produced from molecules needs soil support. So one side vitamins, one side food, one side medicine for treating the disease. This is applicable in plants as well as in human beings. So after oscillation, then transfer of energy, producing of energy, transfer of energy, activation of energy, acceleration of energy, and retardation of energy, and deactivation of energy. Here the deactivation of energy is the term, the homeopathic aggravation. The homeopathic aggravation will come. What is retardation of the energy? The retardation of, I am sorry, the retardation of energy is known as homeopathic aggravation. That's why in Organon it is written that the homeopathic aggravation, if a patient develops, he will be automatically okay without any medicine. But if the deactivation has been done, the homeopathic aggravation will be converted into homeopathic toxicity. That's why if you read Centimetidae medica, particularly in tuberculinum, kali carb, epis mellifica, basilinum, he writes the medicine is double-edged sword, because it may cure when the plant or when the human being pathology physiology is very weak, the pathological disease condition. If we give the similar medicine, this never happens in all the cases. Where the similar medicine is going to act when the physiological condition of particular cell, whether it is human cell or the plant cell, is weak, the soil is not giving support, the human being is not taking any proteins or suffering from deficiencies. In that condition, the energy that was communicated by medicine will not be deactivated by the plant or by the human beings, and that is going to produce the drug disease, and where the drug has to be withdrawn. We cannot call it as a deactivation or something. The drug disease that is more dangerous, the retardation will takes place automatically. that is known as homeopathic aggravation, but the deactivation may takes place, may not takes place. So patient may surrender to the drug disease, or the plant may surrender to the drug disease. If the plant energy has been increased, means the plant that is having strength gained from the soil may not be sufficient plant, and the roots will dry up and the plant will die. This is known as deactivation. This is known as drug disease. And retardation means homeopathic aggravation. See, these all are immunologically observed. That means the homeopathy is totally scientifically understood. But the problem is how to select the similar medicine very easily, how to make it reproducibility. So these are the two difficult conditions for research. We may come out with some conclusion later. So, what is this? This is, in conclusion, life cycle of the plants. What is the advantage of research on plants? What is the difference between research on animals or human beings? The life cycle of the plant is between 100 days. That means more than 3 months to 200 days. Within 6 months, the seed, the germination, the plant growth, and fertility, and everything of a plant cycle of the life is over within 6 months, whether it is paddy, whether it is wheat, whether it is cotton, except for trees like mango trees, where the germination takes place, flowering takes place from a minimum of age of 5 years, and the plant will survive for about 200 years. Whereas the carrying out research on human beings, the human being lifespan is around 100 years. So the lifespan will be going on changing. See, one is childhood, second is after childhood pediatrics problems, then comes the puberty. After the puberty, there will be adulthood. After the adulthood, there will be accumulation of the fat, then geriatric problems. Whereas in plants, it won't be like that. There won't be, see, if we carry out research on, see, we are carrying out one of the research on rose flowers, rose petals, where the flowering, where the color has to be increased in rose, they have to be more pink and more attractive and shining. So the genetic changes have been found out and the medicine has been given. See, this is for a healthy rose plant, making them to flower more and making them to shine, and less red color, more what we call pink color. So that has to be changed, means we have to change the DNA of the plant, and that has been taking place. The studies may come out very easily. The in vitro studies have been found to be highly successful. In the same fashion, the life cycle, the plant mechanism is very easy, and the study will be completed within 6 months of the time. So, in the same fashion, whereas human beings, see, the puberty age is different, the energy levels are different, the blood sugars are in plant, it is not like it is time bound. So, this is the advantage of the doing research on the plants. And safety compounds, see, we need not take informed written consent. The ethical committee will not ask us, because they are plants. So in human beings, we have to see that their health has to be protected. They should not develop any complications during the experimental stage, whereas in plants it is not like that. And the rate, and there won't be any called as placebo effect of homeopathy anymore. That is an old theory. The placebo, whoever is having mind, they will have placebo effect. But who don't have thinking power, or who will not change with brain, they will not have placebo effects. The plants are as natural, as ignorant as human being. They are like child, they only give. In the same fashion, animals, they have mind but they don't have brain. They have mind, they have expressions, but they don't have neurological system. So this is the advantage, but at the same time the pathology is same for human beings as well as plants. Now the present study indicate that homeopathic drugs are able to trigger genes to activate, accelerate, retard, and deactivate. See all the four stages. Hahnemann, when he has written his organon, he do not know that immunology will be understood by a person like me, and he has written a organon of medicine. The activation, that is first oscillation, oscillation into energy, energy into activation, activation into energy acceleration, energy acceleration into retardation, retardation to deactivation. That is number one. Drug will produce the disease, disease will have its own natural causes. When we give drug, it will take oscillation, and the oscillation will be activated. The activated oscillation will accelerate, and the accelerated will be energy will be transformed. If the energy is more than requirement, it will produce homeopathic aggravation. The retardation will take place by the human body or the plant, and the plant will recover, human body will recover from the homeopathic aggravation. If the deactivation process is not up to the mark of the plant or human being or the animal, there will be a drug disease and corresponding to genetic process. This retardation, this acceleration will have enormous effect over the DNA of the human body, and will produce genetic modifications. And this was studied totally on cotton seeds, and the germination has took place. So how they have been done? Some practical recommendation for the use of homeopathic remedies. What we did is, now after doing studies, some of these, whether you don't need so much of elaborate research, but at the same time you want to apply as a general physician to the this agro homeopathy. So some of the medicines: silesia, slow growth, attack of fungi, interpretation of growth, delay in production. See, the germination, fertility is not taking place. So apply silesia 1 is to 1000 dilution. Take any one potency which you think suitable, depending upon the crop production. Suppose it is wheat, a 30 potency, or if it is rice, you kindly take 200 potency, whatever you think best, depending upon the disease condition as on that day, and take the potency, dilute it with 1000 ml of water, spray and give water to the roots. And carbovige, after it have defoliating insects, water deficiency. See, when the water deficiency is present, there won't be any oxygen supply to the plant. Plants are dependent then totally on oxygen, and the oxygen comes from water. Water, the fertility, the Krebs cycle will have enormous effect over the soil, and the Krebs cycle changes will take place because of water, and that water will supply oxygen to the plant. That oxygenation will give strength to the plant and the growth of the plant, size of the plant. And these are from carbovige, because it will also will be used as pesticides. Then there are many medicines: apis mellifica, calcarea phosphorica. See, calcarea phosphorica is phosphorus as well as calcium. When you put into plant into any potency, the plant will grow. So there will be another medicine, magnesium, magnesia carb. You all know that the magnesium that will be supplied. See, the plant also receives. There is one biological
Hi, doctor.
relationship. Sir, over, sir. There is one biological
Okay.
relationship between plants and human beings. The end product of the plants are energetic source of human beings, and the end products of the human beings are any animal are a energetic source for plants. This is known as biological dependency and biochemical process, and it is living together. So usually urea, ammonia, excreta products, these all will be a carbon dioxide, they all will be taken by plants and they grow much. Apart from this, for many fungal infections, the plants need medicines. So there are many medicines you can study. And this is the last slide, the scope of homeopathy for human. See, electronically charged waves from vibration and electronic magnet waves from electronics, enlarged variations of the gene. And the last slide is, this is acceleration growth of cotton. See, you can see the slide, medicated plant and control group, that is placebo group. So the placebo watering, placebo water spraying has not given, that is red mark part is there, that has not grown up, whereas the medicated plant has grown up. This is the modern, this is the latest study, and the agro homeopathy so far is having so much of these studies. And apart from this, these studies, so far as on today, nothing more has been marked, but in vitro studies of immunological changes have been noticed. Thank you very much, sir. Any discussion with you people, I will be very happy, sir. Sir, over, sir.
Okay. Okay.
Thank,
We have
thank
only
you
three
very much
minutes.
to Ganesh, Manoj, and Dr. Kurup. Sir,
Okay, we have only 3 minutes remaining. If anyone have doubts, you can share here. Only 3 minutes remaining.
Definitely no doubts of doubt about the quality of or competency of Dr. Prakash Rao is a wonderful and splendid topic in the IFPH platform. Definitely congratulations, sir. You continue your endeavor. You will get the coming future another prize, no doubt about it, sir. Thank you.
so sir, if anybody says the homeopathic drugs action is not known, you kindly ask him to discuss with me. Now it is the pathways of homeopathic medicines have been understood.
Yes, good.
So the mechanism of action of medicine has been understood. And there is a seminar that is going to happen only on these theories. And the study has been conducted by me on immunology. So now the mechanism has been understood, sir. Now homeopathy is a totally scientific system of medicine. The only drawback is how to select a medicine, how to make prescription easy is not possible. It will come. It's possible. Everybody is doing their hard work. Of as a physicians, you are treating various cases, so that hard work has to come down, basing upon the scientific studies, sir.
Okay, sir. Thank you. We have to go for the next session, sir. Yes, Dr. Ganesh, please.
Yes, definitely. It was a very good session indeed. And as rightly pointed out by Dr. Manoj, we need to discuss about these things, and we are ready to bring those persons who are having any doubt on the subject. Definitely will come to you, sir. You can make all these things, and we need some more presentation of this calibre category. Congratulations, sir. It's over to to Mr. Shailu Rajan to conclude the session.
Okay, now we are inviting to Mr. Bob Lim for vote of thanks.
Yes, Dr. Babu.
Dr. Bob Lim.
Yes, Dr. Babu. Yes, yes, yes. Yes, please.
Maybe sleeping. It's midnight there.
No, I know he's always there. Yes, yes.
No, no,
Please
no.
unmute.
Not responding.
Yes, anyway. Then I shall. Anyway, I'm happy to say what a thanks to this session, and thank you, Dr. Prakash Rao sir, for such a wonderful session in this evening. We hope some more presentation will be there on the subject again, and so that homeopathy will reach every home with this with the no, with the same intensity what it is now. Thank you very much, sir, once again. Thank you.